the topic of our project is protein and peptide drugs, challenges and innovation for using therapeutic process. This project will be presented by me, Aizhan, by Nazerke, Inabat and Angsar. Here you can see our table of content. First one is introduction. It will be general information about peptide and protein drugs, challenges in using innovation in drug development, real-life successful examples of the peptide and protein drugs, and the conclusion and our references. We should understand what peptides and proteins drug. Peptide drugs are composed of short chain of amino acids. It is typically less than 50 and have a simple, most linear structure, while protein drugs are larger molecule with complex structure. Peptide drugs often act as signals hormones in our body, and while protein drugs, it is an enzyme and antibodies in our body. Peptide drugs are less stable and they are quickly broken down by enzymes. And the bus type typically require injection for administration. Here you can see the general information, what I said, the differences between peptide and protein drug. Now I would like to share with one experiment in which scientists made medicines called GLP-1 agonists to help people with diabetes and obesity. These medicines help the body to make more insulin and reduce hunger. In 20 and 21 years, in 28 September, the company that called Nestle did a study to see if drinking a little bit of special protein before meals could help people with diabetes control their blood sugar. They asked 26 people to drink either a protein drink or plain water before eating. After 1 week, they switched the drinks. The result showed that people who drank the protein had 22% lower blood sugar after eating compared to those who drank water. Their GLP-1 hormone also increased, which helped their bodies process blood better. And here we can conclude that protein helps the body to better control blood sugar level and improve the absorption of food in our body.
So now I'll talk about the instability and short half-life. The peptides and proteins are inherently unstable in the human body, leading to rapid degradation and a short half-life. And this instability necessitates frequent dosing, which can be inconvenient for patients. To address this, researchers are exploring various modification strategies to enhance stability and extend the half-life of these therapeutics. And the next topic is bioavailability and delivery challenge. Bioavailability and delivery challenges. The delivery of peptide and protein drugs is complicated by their limited bioavailability and challenges in crossing biological membranes. Advanced delivery systems such as protein nanoparticles, PNPs, have been developed to improve the pharmacokinetic properties of these therapeutics. PNPs often offer high biocompatibility, biodegradability, and low toxicity, potentially overcoming issues like high bioavailability and high toxicity associated with synthetic nanoparticles. And however, clinical translation of PNPs is limited with only a few formulations approved by the FDA, such as Abilgazyuly and certain viruses like particles VLPs. Challenges include in vivo interactions that may affect safety of functions such as protein conformation changes and potential inflammation. And our next topic is about high production costs and synthesis complexities. High production costs and synthesis complexities manufacturing peptide and protein therapeutics is often complex and costly, limiting their widespread clinical use. And the synthesis of PNPs presents challenges in drug encapsulation and pharmacokinetic variability between batches, affecting clinical outcomes and patient safety. And additionally, large-scale production faces limitations due to higher costs and lower yields, hindering broad clinical adoption. And our next topic is immune reactions and potential side effects. Immune reactions and potential side effects. Immunogenicity is critical concern for peptide and protein therapeutics, as the body's immune system may recognize these molecules as foreign, triggering immune responses that compromise therapy. And in vivo interactions between PNPs and the biological environment can jeopardize their safety of function, leading to potential inflammation and other adverse effects.
So the next part is innovation and solutions, and we will discover the improving stability by the molecular modification, new delivery methods of drugs, and biotechnological methods. So methods for improving stability. The first is pegylation. Pegylation is the attachment of polyethylene glycol to peptides or proteins to enhance their pharmacokinetic properties. And pegylation interferon is an example of PEG drugs. And the second one is cyclization of peptides. Cyclization involves the linking peptide sequences to form a loop, stabilizing their structure. And here are the three types of the cyclization. The first is head-to-tail cyclization. In here, they are connecting the N terminal to the C terminal, and the second is backbone to the side chain cyclization, is linking backbone atoms to the side chains. And the third one is side chain to side chain cyclization, is connecting two side chains to create a loop. An example of this is a celepressine, is a modified form of vasopressin with a cyclized structure. And the third one is substitution with a non-natural amino acids. It's about replacing the L amino acids with the D amino acids to resist the enzymatic degradation. And the fourth one is a side chain modification. It's about modifying peptide side chains can enhance the binding affinity and selectivity and stability. And the example of this is a semaglutide and liraglutide have fatty acids modified side chains which increase their stability and half-life. So the next page is new delivery methods, and here the first is liposomes. Liposomes is a spherical vesicles made of the phospholipid bilayers, and it protects drugs from the enzymatic degradation and improves the targeted drug delivery. And the example of this is a Doxil. And the second one is solid lipid nanoparticles, or another name is SLN. And the definition of this is a nanoparticles with a solid lipid matrix where the drug is dispersed in an amorphous crystalline lipid core. And it allows the controlled drug release over time. And the example of this is the insulin-loaded SLN for oral drug delivery to improve absorption. And the third one is NLC, or nanostructured lipid carriers, and is a second generation of lipid nanoparticles containing a mixture of solid and liquid lipids. And the difference from the first one is have a higher capacity than SLN. So example of this is NLC-based calcitonin delivery and is used for osteoporosis treatment, which improves the bioavailability. And the second page is a biotechnological methods. Biotechnological methods is about isolating bioactive peptides, and it's about the peptides are derived from the natural sources like animal, tissues, plants, or microbes. And the second one is non-ribosomally synthesized peptides. Unlike from the normal protein synthesized which uses by the ribosomes, some peptides are produced by the enzyme complexes known as a non-ribosomal peptide synthesizer. And these peptides often have a unique structure that makes them more stable and bioactive. And the next is venoms and toxins. Venoms are toxins also used in drug modification, and venomous animals like snakes, scorpions, cone snails produce the bioactive peptides that target the ion channels and enzymes and receptors. And many have a high potency and selectivity, making them good drug candidates. And the next is enzymatic synthesis. It uses the enzymes to assemble short peptides in the controlled manner, mainly applied for food additives, pharmaceuticals, and industrial chemicals. And the next is fermentation. Fermentation is a biotechnological approach where microbes produce the bioactive peptides. It's eco-friendly and cost-effective for large-scale production. And the next one is recombinant DNA technology. It's used to genetically engineered bacteria, yeast, or mammalian cells to produce the peptides and proteins. And the last one is genetic code expansion. It expands the natural 20 amino acids repertoire by introducing the non-natural amino acids.
So hello guys, I will explore the topic about the real examples of peptide and protein drugs. So first, and a real successful example is about insulin. Insulin is a hormone that regulates the blood sugar level, and it treats diabetes type 1 and type 2. In the past, insulin was taken from the animals like pigs and the cows, and nowadays, thanks for the biotechnology and the genetic engineering, insulin is taken from the E. coli bacteria, and the insulin modern insulin has better features like more stable, last longing, and making it easier for patients to take. About the growth hormone, or another name is somatotropin, is a protein hormone that controls growth and the metabolism. If kids don't have enough growth hormone, they may not grow, and in the adults, if there is not enough level of growth hormone, they will have trouble with metabolism. Now, with recombinant technology, we can make synthetic growth hormone, making it more available. Besides medicine, it's used in sport drugs and help with aging. The next is about peptide antibiotics. It's play a major role in immunology. Peptide antibiotics are a special type of antibiotical drugs that attacks the bacterial cell walls. A well-known example is valnamicin, which fights bacteria that resist another antibiotics. Unlike regular antibiotics, peptide antibiotics don't often lead to resistance, making them promising straight to fight bacteria infections. I will explore about the insulin treatment, their influence to the human health. So we know that insulin therapy is a key way to treat diabetes, and especially for people with type 1 diabetes where it's needed to stay alive. For type 2 diabetes, insulin is work when another treatment is not available or is not successful. Success is measured by the reaching a target HbA1c level less than 7%, which shows blood sugar is under control, and by lowering the risk of problems like eye, kidney, or nerve damage. About the statistics that provided by National Health and Nutrition Examination Survey from the 2009 and the 2020 years, they provide that people with diabetes type 1 that supply the insulin treatment, they achieve 25% of that people achieve HbA1c level less than 7%. And what about the diabetes type 2? Also, the people that supply the insulin treatment also reach less than 7% of HbA1c. About the basal insulin, people with type 2 diabetes that require the basal insulin, only 12% of them reach the less than 7%. The second is about the reducing the risk of microvascular complications. People that have diabetes type 1 that have insulin treatment, they reduce their risk of microvascular complication from 35% to 76%. These data is provided by DCCT. About the diabetes type 2, people that supply the insulin, they go through intensive therapy, 25% of them reducing their risk. So, by the data that I was provided, only a few amounts of people resist against the trouble of diabetes. What did World Health Organization promise to fight against this problem? In 2022, countries in the World Health Organization agreed to make big goals for diabetes. They want to better fight to diabetes as part of the plans to stop diseases that don't spread the heart problems or diabetes. The World Health Organization in Europe and the European branch of the International Federation that helps with the diabetes agreed to work faster to fight these problems. And by 2030, they promised that 80% of people with diabetes get the right diagnosis. Also, 80% of people with diabetes keep their blood sugar and the blood pressure under control. 60% of people with diabetes who are 40 or older get special medicine, and 100% of people with type 1 diabetes can afford insulin and the tools to check their blood sugar by themselves.
So at the end, to be concluded, we want to say peptide and protein preparation have played a revolutionary role in medicine, offering effective solutions for treatment of various diseases. We reviewed the key aspects of these drugs, their features and benefits, problems associated with their use, the latest innovation in the development of these drugs, as well as the examples of successful drugs.
So the future of peptide and the protein drugs promises even more innovations, including the improved delivery methods, synthetic analogies, and the personalized medicine. These developments will help make treatment more accessible, effective, and safe for patients around the world.
Goodbye. Все.